Hello, and thank you for joining us today. I am pleased to welcome you to the TriLink Biotechnologies mRNA Basics webinar series. I'm Daniela Ventro, Senior Marketing Manager at TriLink, and I'll be moderating our discussion. In today's webinar, we'll discuss a key component of every mRNA therapeutic program, analytical testing, taking a look at some considerations and common mistakes to avoid. mRNA Basics is an educational webinar series with a focus on a major mRNA research topic each month. In addition to today's presentation, a question and answer session with our expert speaker will follow. We encourage you to send us your questions during the presentation by using the Questions tab located in the panel to the right of your screen. We will address as many of your questions as we can during our Q&A session. The webinar will be archived on TriLink's website, and we'll send you the link within a couple of days. More webinar topics will be announced soon, so be sure to subscribe to our mailing list for the latest updates. And with that, I'd like to introduce today's speaker, Jessica Madigan. Jessica is a Director of Business Development at TriLink Biotechnologies and has played a pivotal role in pioneering contract manufacturing of messenger RNA. She has served as the manufacturing support lead for groundbreaking programs in mRNA therapeutics. With unparalleled experience and expertise in GMP programs, Jessica has successfully guided more than 30 programs into the clinic. For over a decade at TriLink, Jessica has worked in customer and technical service across a variety of product lines and application areas, including oligonucleotides, mRNA, and small molecules for vaccine development, gene and cell therapy, and protein replacement therapies. She received her Bachelor's of Science in Chemistry and Biochemistry from San Diego State University and is certified as a project management professional. And I'll go ahead and turn it over to Jessica for what's on the agenda today.
Today, I will be focusing my presentation on analytical testing, and the three most important components of that are raw material testing, in-process testing, and then finally, release testing. First, I will cover the basics of mRNA designs and considerations. In order to understand the important quality control attributes, it's first important to understand the basics of mRNA manufacturing. So again, I will briefly touch on this, but for more details, I invite you to view some of our other webinars in the mRNA Basics series. After we cover the design and manufacturing, I will cover the important quality control attributes of plasmids as they are critical raw materials entering the manufacturing process. We will then spend the majority of our time today on a variety of mRNA quality control assays and how we can use these assays to set specifications and support stability studies. mRNA design considerations. When approaching an mRNA synthesis, these are the important key points of concern. Target selection. You choose your protein of interest and potentially screen various codon optimizations and untranslated regions. The 5' UTR is upstream from the coding sequence. Within the 5' UTR is a sequence that is recognized by the ribosome, which allows for the ribosome to bind and initiate translation. The 3' UTR is found immediately following the translational stop codon. The 3' UTR plays a critical role in translation termination as well as post-transcriptional modifications. An N7-methylguanosine cap structure is important for ribosomal recruitment. Base modifications are important in eliciting immune responses. A poly-A tail is also required for ribosomal recruitment, and the purification method needs to be defined based on the required specifications and ultimate scalability of the product. Here is the standard mRNA manufacturing flow. We're going to start with the plasmid linearization step, where we take circular plasmid and digest to a linear template. Then we move into transcription. In the transcription, you will have a variety of proteins and NTPs, including T7 polymerase, RNase inhibitor, pyrophosphatase, nucleotide triphosphates, cap analogs, and potentially modified bases. After transcription, there is a DNA step to digest the DNA template, which is then followed by difiltration. After difiltration, we will embark on our purification, where we have a variety of options available. And then finally, post-purification, we will do another difiltration step and enter final QC. mRNA purification is required to remove salts, NTPs, cap analogs, proteins, and residual DNA. There are also options to enhance the purification methods by removing truncated RNA species and double-stranded RNA. So depending on the method of manufacturing and purification selected, we may need to control for these impurities throughout the process. Now that we have an understanding of the mRNA manufacturing process, I will touch on plasmid quality. Here I am showing a standard quality control release strategy and specifications for a plasmid leading into mRNA manufacturing. It is important to test for process-related impurities, residual RNA, residual host cell proteins, and residual DNA. This will help ensure that the residual impurities are not carried through to the end messenger RNA product. It's also important to look at the percent isoform purity, endotoxins, and sequence verification. Low endotoxin values are required. For mRNA, what you put in is what you get out. The purification methods for mRNA are not traditionally endotoxin reducing. So we recommend at least less than 5 EUs per mg for plasmid DNA. mRNA quality control. Here is a large panel of in-process and final release assays offered by TriLink. On the left side of the screen, you can see a number of messenger RNA-specific assays, and on the right side of the screen are a number of USP-based methods. We'll go into detail on more of these methods today. In general, though, for agarose gel, you're going to be looking at length and purity of your RNA product. This is typically used as an in-process control, as it is a qualitative method. Concentration and UV purity are measured throughout the process in order to quantitate your yield from transcription and monitor throughout the process. RNase and DNase detection can be run on raw materials as well as in-process controls in order to monitor product quality. For final product, conductivity and a functional test are important, as well as capping efficiency, poly-A tail length, residual protein, residual plasmid DNA, double-stranded RNA. Purity is offered by a variety of methods, including fragment analyzer, bioanalyzer, or ion pairing reverse phase. And finally, identity is also offered by a number of methods: sequencing, fingerprinting, and RT-PCR. We'll go into more of these details on each one of these methods in the following slides. Capping assay. In order to assess the capping status of a messenger RNA, which is actually quite challenging because we are looking for the addition of a single methyl group to an RNA that can be millions of Daltons in length, we developed a capping assay at TriLink in which we've cleaved the 5' end of the transcript, and then we use an LCMS method to detect for the heterogeneity of the 5' end. On the right-hand side of the screen, I have two examples of a capping assay. One is an enzymatically capped product that has not gone to completion, and the other is a clean capped transcript. LCMS characterization of poly-A tail length. In order to assess the poly-A tail length, we have developed another LCMS method. First, we digest the messenger RNA product, leaving the poly-A tail intact. Then we run an LCMS. As you can see here, we take the deconvoluted spectrum and relate that to the intensity to determine the poly distribution. A residual protein assay measures the amount of residual protein present in messenger RNA samples after purification. Typically for final product, residual protein is measured using a nano-orange protein quantitation assay. Silver stain is highly sensitive, but is a qualitative assay in nature. And we would normally only use silver stain to optimize protein clearance in process, and we recommend using the qualified and quantitative nano-orange method for final release. Residual plasmid DNA assay measures the amount of residual plasmid DNA present in mRNA samples after purification. This method uses a dual-labeled probe and a real-time PCR reaction assay to measure the amount of amplicon present in mRNA samples by comparison with the standard curve generated from a known amount of input plasmid. We have developed at TriLink platform detection methods for residual DNA against kanamycin resistance, ampicillin resistance, or generic TriLink UTRs. Custom primer and probe sets can also be designed as well for specific sequences. Double-stranded RNA can be assessed by slot blot. At TriLink, we use a J2 antibody for this method. This method is considered qualitative and can be made semi-quantitative at best through densitometry of the blot. But the slot blot assay is notoriously challenging for inconsistencies from blot to blot, and it is recommended to develop an interferon response assay, like a THP1 dual labeled cell assay, in order to correlate slot blot data to interferon response. For assessing the purity of a messenger RNA, we offer a variety of different methods, including bioanalyzer, agarose gel, fragment analyzer, and ion pairing reverse phase HPLC. Fragment analyzer is the most common method used for final release. A fragment analyzer is used to establish the RNA integrity using an Agilent fragment analyzer instrument. This system uses an automated parallel capillary electrophoresis to provide quality control for nucleic acids. Purity is established by smear analysis as compared to a reference RNA. Ion pairing reverse phase HPLC was derived from its parent technique, reverse phase liquid chromatography. It uses a hydrophobic stationary phase and a mobile phase with varying hydrophobicity, depending on the ratio of polar and organic solvent. It is an effective method for evaluating the integrity of the RNA. In this slide, I am showing a firefly luciferase messenger RNA that has been purified by oligo-DT. For the fragment analyzer, the blue traces material after the first difiltration, but before purification, and the red trace is after oligo-DT, and the black trace is final product. This is how we can monitor purity throughout the manufacturing process. For the IPRP HPLC example shown on the right, the blue traces the material before difiltration and before purification. The black trace is after oligo-DT, and the pink trace is the final product. The IPRP HPLC method does offer baseline resolution of the tailed and tailless mRNA and is an easy way to assess the purity of the messenger RNA product. Identity testing. For identity testing, the most commonly recommended method is RT-PCR, followed by PCR, followed by sequencing. This can be done with 4-fold coverage, 2-fold in each direction, and the results are reported as alignment to consensus sequence. There are also a few other options to note, which would include the RNase digestion assay, in which digestion of the mRNA product is carried out by RNases, and then we run an agarose gel to visualize the complete digestion of the product, confirming RNA identity. In vitro translation assay can also be achieved using a cell-free wheat germ extract translation assay with a BODIPI labeled tRNA. The resulting product can then be run on an SDS page gel, and the size of the protein can be determined against the marker, as shown in the example here on the right side of the screen. RT-PCR for a specific amplicon can be done as a singleplex or multiplex assay. The results would be a detection of a desired amplicon or amplicons by agarose gel. Setting specifications. Developing specification for the manufacturing of an mRNA require a comprehensive and critical analysis of the requirements that are used in developing and validating sound analytical methods. The approach to setting specifications for an mRNA are not straightforward, as the molecules are very complex. Because of this, it's important for CMOs and their sponsors to have process-driven approach to setting specifications that meet both the FDA and the ICH standards. At each stage of development, from research to preclinical to phase 1 and beyond, after securing FDA approval, companies need to ask themselves what's absolutely necessary to ensure safety and efficacy of their product. Ultimately, the customer will define what specifications must be met in order to maximize the likelihood of regulatory acceptance. It's also important to note that amounts required for testing and release and retention samples may actually exceed the amount of material you need for your clinical studies. So you need to start thinking about what release assays are needed early in your program in order to plan for the eventual manufacturing scale needed for production. Now we're going to move into some example specifications The first shown here is that a final QC test is identity. Again, sequencing is the most common method. Our specification would be that it conforms to the theoretical sequence. We also would, important to include, is visual appearance. We would want a clear, colorless solution that is free from visible particles. Agarose shell can be run to visualize the RNA length as compared to a reference ladder, and we would want the migration of the RNA to align with the known theoretical length by the ladder. RNA content is done by UV absorption at 260. We're typically going to target the target concentration plus or minus 10%. pH of messenger RNA is going to be driven by the buffer, but majority of RNA sequences can be found in the pH range of 5 to 7. Osmolality can be important, especially for ex vivo applications. And a messenger RNA packaged in water would generally have an osmolality of 0 to 50 milliosmoles per kilogram. For fragment analyzer, we would be setting the specification for purity based on the results from each phase of manufacturing, from research to preclinical and then ultimately clinical trials. It is important to note that the length of the RNA is going to be directly proportional to the achieved integrity. The longer the sequence, the harder it is to achieve high purity. With oligo DT, we can typically drive purity levels to greater than 70% for most constructs. Bioburden should be driven by your dosing strategy. Less than 1 CFU per mil is typically achieved. Endotoxins also should be driven by your dosing strategy, but less than 3 EUs per mil is very common for messenger RNA applications. For residual proteins, we will be looking for less than 2% weight to weight. For residual plasmid, we would be looking for less than 1,000 picograms of DNA per milligram of RNA. Residual solvent panel will be chosen based on the manufacturing process, and so TriLink can have a conversation with you around what residual solvents are important to test for. In a standard panel, we're going to look for any residual solvents that are going to be known safety standards, such as acetonitrile, chloroform, acetic acid, or ethanol. For capping efficiency, we would be looking for greater than 90% capped. For slot blot, we would report for information only, the image, and important to control for lot-to-lot variability. And finally, identity shown here, we could do the in vitro translation assay, which is the SDA's page gel to show the protein of a particular size has been made by the messenger RNA. Now, following release of your product, don't forget that you also need a stability study in order to file your IND. Typically, 1 month stability is absolutely required, and 3 months is preferred. TriLink can assist with executing a variety of assays at storage temperatures and time points, and we're happy to discuss with you what's the best fit for your program. Management of your stability study would include project management services, coordination of internal and external release assays, ICH compliant temperature storage chambers, and a quality assurance approved certificate of analysis for reporting all results at each time point. TriLink has committed to meeting the growing needs of our customers and the mRNA industry. We have a 118,000 square foot facility in San Diego, California, with expansion opportunities. In the last year alone, we have added 10 additional ISO-classified clean rooms to support increasing demands and added plasmid manufacturing services. Thank you for your attention today. I hope I have provided you with sufficient information on important considerations for analytical testing for mRNA therapeutics. Now I would like to open the webinar for the question and answer session.
Thank you for that presentation, Jessica. The audience has been actively submitting questions, so let's get to them. Please feel free to continue to submit questions. Our first question, how do I know which analytical tests I have to include?
Hi, Daniela, thanks for that. The analytical tests that you need to include are going to be driven by your discussion specifically with the regulatory agencies. Most important, you're going to want to include a potency test, a purity test, an identity test, and safety tests. So again, potency, you're going to be looking for what the concentration of your messenger RNA is. Identity can be done by sequencing. The purity can be done by fragment analyzer, and then you would look for endotoxins and bioburden. Those are going to be the most important ones to include. Again, we have a large, extensive menu, though, beyond that to best characterize your molecule, and we're happy to have discussions with you on what's going to work best for your program.
Thanks, Jessica. Next up, can I order analytical testing as a standalone service from TriLink?
Currently, we do not offer analytical services as standalone testing. Right now, we're really focused on supporting our custom manufacturing services, and we really see the analytical services as an add-on support to our clients. So unfortunately, right now, we don't offer standalone testing, but always feel free to reach out to us if you have a specific need or program. We're happy to consider it, or potentially, refer you to a third party that may be able to help with that service.
Thanks, Jessica. How much material is needed for analytical tests?
That's a good question. I would say that for, and I'll scroll back to kind of the release panel we previously shown. This year, the most material is going to be consumed by the bioburden test, and it will be driven by the specification. So, for example, on this panel, we've shown that the specification is less than 1 CFU per milliliter, which would mean we'd need to test 1 milliliter of product for aerobic bacteria and 1 milliliter of product for fungus. In the case, though, that you're testing for a specification of, say, less than 1 CFU per 10 milliliters, now you'll need to test 10 milliliters for aerobic and 10 milliliters for fungus. In addition to that, you also have to perform suitability testing, which contains about 7 times the amount needed for regular testing. And then you also have to consider what your concentration of your product is. So kind of best case scenario for bioburden, at a less than 1 CFU per mil, you're consuming 9 mgs of product at a 1 mg per mil concentration. Now, as you increase your stringency on your specification and you increase your concentration, you're going to really drive up the consumption of that product. Endotoxin testing typically only takes about 1 milliliter. And then the other huge consumption is usually in the residual solvents. Depending on what parts per million you're testing down to and what system you're using for GC injection, you could probably expect to consume another, half a milliliter for each solvent that you're testing for. Identity testing, obviously sequencing takes very small amounts of material, maybe 10 microliters. Residual plasmid will probably take about 100 microliters. Capping efficiency is also variable depending on the length of your product. So for capping efficiency, we're going to be looking for a certain number of moles of product on the 5' end of the molecule in order to detect by the LCMS method. So the longer your messenger RNA is, the more material you'll actually be using in the capping assay. For a 1,000 base pair messenger RNA product, you probably only need about 500 nanograms, 500 micrograms of product. And for each 1,000 bases, you'll double that. So for a 2,000 base pair, you'll need about 1 milligram of product.
Thank you, Jessica. Next question is, why is sterility not on the release panel?
Good question. So sterility is not included on this release panel because at TriLink, our facility is a low bioburden facility only. We are producing non-sterile APIs. Our manufacturing process for GMP production is carried out in an ISO-classified clean room, and the final manufacturing step is a 0.2 micron filtration in an ISO-classified space. We do use aseptic practices in order to minimize any contamination, but we have not done fill validation. So In order to claim sterility, the process must be validated as sterile, and that is not included in our services here. And so really low bioburden is what we would recommend for the release panel.
Thank you. Next up, do methods need to be qualified for release? And if so, how long does method qualification take?
Yes, methods absolutely should be qualified for release. And so with respect to the USP methods, we would of course make sure that your product is suitable to be run by that USP method. But for non-USP methods, we highly recommend that we do a method qualification specifically for your product. And so at TriLink, we can do method qualification per the ICH Q2 guidelines. We would basically agree with you on which characteristics of the validation need to be executed. We would be testing for accuracy, precision, specificity, limit of detection, limit of quantification, and linearity and range. And so those are all going to be tested depending on, obviously, if it's a quantitative method or not. There may be certain parameters that we would exclude from the testing, but in general, we would always recommend that your purity method by fragment analyzer go through a qualification. We would recommend that the residual protein method be qualified, the residual plasmid method be qualified, and your concentration assay be qualified. Now, for the other assays, such as capping, the double-stranded RNA, or identity testing, those methods are very difficult to qualify. The capping method itself would require synthesis of some standards of your actual capped form, and making those standards is actually very challenging. And so it's something where we typically would proceed with a characterization only report of results for phase 1, with the intention of moving forward to a qualified method for later phases. For identity testing by sequencing, we can offer qualified methods for that, but we do outsource that to a third party, which would all be managed by TriLink.
Thanks, Jessica.
And sorry, I didn't.
Go ahead.
I'm sorry, Danielle, I didn't answer the second part of the question of how long does this take. So usually we would allow at least 8 weeks, preferably 10 weeks for method qualification. And you obviously have to have material available for your program to do a method qualification. So when contracting with TriLink for a GMP program, we would manufacture materials under an engineering run, or some people refer to this as a toxicology run. That material that's produced would then be used for animal studies to support your IND filing. And a portion of that material would be used by TriLink to complete the method qualification. Then once methods are qualified for release, you would produce your GMP material and then release using the qualified methods. So again, that process usually takes about 8 to 10 weeks to complete once material is available to pursue the method qualification.
All right, great. We might have time for just 1 or 2 more questions here. Next up, what type of documentation is provided with an RNA product? For example, a C of A or COO for any materials of biological origin used in the manufacture?
For our GMP products, you're going to get a lot of documentation. In fact, for GMP, the documentation itself is almost as important as the product. You will be receiving the full executed batch records, all of the raw material, C of A, COOs, and release from TriLink's raw material group. The final product will have a certificate of analysis reporting all results with the specifications and then approved by TriLink Quality Assurance. You'll be receiving a certificate of origin certifying that the product is going to be limiting the risk of BSE or TSE, and you're going to be receiving a certificate of release, which certifies that it's been made to GMP standards, specifically ICH Q7 section 19. In addition to that paperwork, for your toxicology studies, you would be receiving a COO and a C of A, and Yeah, I think that pretty much wraps it up.
Great, thank you. And unfortunately, that's all we have time for today. Thank you for taking the time to join us and for your questions. I'd also like to thank our speaker, Jessica Madigan. As a reminder, the webinar will be archived on the TriLink website, and we'll send you the link within a couple of days. Have a wonderful rest of your day, everyone.